All material in pistil-pollen interaction following the pollination. In 1973, Cass had shown that two sperm produced by male gametophyte remained together for considerable time. Later, several workers demonstrated that the vegetative nucleus also remains in contact with a sperm cell and three move through the pollen tube as a male germ unit. These brought to light the phenomena of sperm of dimorphism and suggest that fate of two sperm delivered by pollen tube may be predetermined. Knox and his associates presented a computer-assisted three-dimensional structure of the male germ unit of Brassica compratis. Credits go to McCockney et al. 1985 for their paper and G. Mays McCockney et al. in their paper published in 1987. It is now clear that at least some plants the inheritance of extranuclear gene is biparental. In 1973, Cass reported separation of female cells from the vegetative cells. Cytoplasm of barley pollen. However, Russell in 1986 was first to obtain an enriched fraction of purified sperm. Since then, several protocols have been described for isolation of large number of live sperm by bursting of pollens and pollen tubes in variety of angiosperm. Possibility to manipulate isolated sperm has given birth to new field of pollen biotechnology. There is a book on it written by Shivana and Swamshane in 1977. Techniques have also been developed to isolate live embryo sac and egg cell, demonstrated by Hishino et al. In their paper. For long, suspensor was regarded as an ephemeral structure of not much importance in embryo development. However, a close examination of the physiological, biological, and ultrastructure feature of this structure has established beyond doubt that their early ages of embryogenesis suspensor is metabolically more active than the embryo proper and plays an important role in nutrition of the pro-embryo. Currently, several scientists are investigating the genetics of embryogenesis in angiosperm to identify the gene involved in events such as pattern formation, cell and organ differentiation. During embryogenesis, classical approach of screening developmental mutant defective in normal embryo development is being followed. Asexual development formed without pollination and fertilization apomixis have been known for over a century. Since apomatic offspring are genetically identified to the mother plant, it offers an inexpensive way to clonally multiply selected lines, fairly large number of agronomic crops. Embryological studies have revealed the origin of development apometic understanding genetics and apomixis involved in the modern method of molecular biology so that crops can genetically modify for apometic trait. This has been demonstrated by a paper written by Kush et al. And another paper by Savidan in 2001. Here I also want to show you the picture of Joseph Hassan Harrison. Experimental embryology has long history. As early as 1904, Henning had invented work on embryo culture. In 1925, potential of this technique is increasing in success rates of hybridization programs was demonstrated by Liebeck in 1925 and 1929. Since then, ex avulo and in avulo hybrid uh, embryo culture has enabled raising several rare hybrids which normally fail due to premature abortion of hybrid embryo. Until recently, it was not possible to culture isolated pre-globular zygotic embryo. Therefore, in crosses where abortion occurred in an early stage of embryo rescue was possible only in avulo embryo culture. In 1993, Liu et al. Achieved complete embryogenesis in the cultures of excited 8-cell pre-embryo mustard by using rich nutrient medium and modified culture technique. At almost the same time, Crange and Lorage (1993) and Holm et al. (1994) succeed in raising fertile plants from in vitro and in vivo formed zygotes of maize and barley, respectively, by supplementing the culture medium with nurse cells. In early 1960s, embryogenesis developed the technique of intra-ovulation, ovarian population, and test tube pollination by Katna 96 to widen the scope of hybrid sexualization by passing the barriers of crossability for which reaction occurs on the stigma or style. By 1990s, methods were available to isolate large number of living male and female gametes of angiosperm, and studies have been initiated to manipulate the naked gametes. In 1993, Crange and Lorage succeed for the first time in regenerating full fertile plants of maize from zygote produced by in vitro fusion of egg and sperm cells. This report in vitro fertilization has opened up in a new approach to crop improvement. In 1958, it was reported for the first time that somatic cell can form embryo like zygotic. Since then, somatic embryogenesis has been observed in over 100 species. Embryogenic cultures of carrot are being used to clone embryo-specific and embryo-enhanced genes. The possibility of producing millions of somatic embryos within a short period offers opportunity to explain the phenomenon in clonal multiplication plant commercial scale. Artificial produced by encapsulating somatic embryo in a suitable jelly would allow mechanical showing of somatic embryos in the field. It was established by Redenbaugh in 1993. The role of haploids in breeding and genetics of higher plants has been emphasized for considerable time but restricted ability of such exploitation. In 1964 and 1966, Guha and Maheshwari demonstrated the possibility of raising large number of haploids from pollen grains. At the androgenesis of the Datura inoxia by culturing immature anthers. To date, anther pollen culture has enabled raising haploid plants or cell lines in over 200 species. In many plants, it has been possible to raise androgenic plants by isolating microscope culture, which offers many advantages over anther culture. Androgenic haploids are being used to produce improved cultivars of crop plants. The limitation of anther or pollen culture in producing viable haploids of some species prompted exploitation of the alternative source of haploid cells, female gametophyte, the first reports of in vitro gynogenesis in Hordeum vulgare was established in 1979 by Sannonium. Since then, gynogenetic haploids have been raised through ovary or ovule culture in at least 23 species. Manipulation of embryological process has been attempted through genetic engineering. Mirani et al. In 1990 developed a widely applicable method to induce male sterility in flowering plants. It involves inserting a chimeric gene comprising a bacterial gene coding for ribonucleose and nuclease and a tapetum-specific promoter. More recently, pathogenic parthenocarpic eggplant has been produced by introducing an auxin-coding bacterial gene. There are some suggested reading or what we can references. These are list of references here. That's all for the chapter 1.